Welcome to the Gene and Cell Therapy lecture. Today we're going to talk about pluripotent and somatic stem cells, so we're going to have a focus on stem cells today. And what are we going to talk about? We're going to talk about, well, what is a stem cell, some definitions, classifications of what the stem cells are, assays, because we want to know why do we know what a stem cell is, tissue stem cells, and the therapeutic use, because we're going to talk about during the whole lecture about gene therapy and cell therapy, so it's the application of all. So what is a stem cell? Well, you can simply look at this scheme. You have a niche, something which interacts with a cell, which is per definition a stem cell. This stem cell can either produce more of itself, expand by symmetric division, or maintain itself, meaning that one cell stays a stem cell and the other cell differentiates and becomes specialized, taking over a specialized function. Or, after division, both cells would differentiate and get lost as stem cells, but become something highly specialized at the end. So, in both first cases where we have either expansion, symmetric division, or maintenance of the stem cell, asymmetric division, self-renewal happening. And of course, you want to maintain your stem cells or even expand them, otherwise the organism would disintegrate. So to do it, to explain it a little bit more, well, fancy maybe, or easier, just imagine that the United Kingdom would be a stem cell. Okay, that cell can be self-renew, it stay what it is. Yeah, that would be the case like the United Kingdom stays together. Or it can fall apart, let's say differentiate towards single units which have different functions, different identities, and one can easily imagine how that works in the United Kingdom, especially when thinking of Scotland. So certain units. So how is all this controlled? Well, we have in a stem cell intrinsic and extrinsic mechanisms controlling if the stem cell differentiates or stays what it is. So the intrinsic mechanism would be, for example, the parliament. In a cell it would be the nucleus, having transcription factors in there, a transcriptional program ongoing, telling or forcing the cell to stay a stem cell during division. We have a niche, we have surrounding cells there, it would be the European Union, for example, here, to tell the stem cell to stay a stem cell, influencing it by, in this case, political means. Stay what you are, stay united. And that's in case the niche, like in the bone marrow for hematopoietic, so blood-forming stem cells, usually the niche, the close niche, tells the stem cell to stay a stem cell. But we also have signals coming from far apart in the body, in this case from wherever, telling the stem cell to differentiate, to fall apart, to become specialized. That could be in a body like an inflammation somewhere ongoing, telling the bone marrow, well, we need more immune cells. Stem cell get activated, differentiate, and produce more immune cells to fight an infection, for example. So how do we classify stem cells? We have different potencies. We can have a totipotent stem cell, which is basically the fertilized zygote. These cells can become everything in the body, all cells, all tissue types, including the placenta when it comes to mammals. We have so-called pluripotent stem cells. These stem cells can differentiate towards or can form all cells of all germ layers, of all three germ layers, but not the placenta, for example, or at least parts of the placenta. So only all tissues of the body. We have multipotent stem cells, which are basically tissue stem cells, producing cells of a certain tissue, and unipotent is usually the term which is not really used. These are just specialized differentiated cells. So when we look at totipotency and ask the question, well, how long is a zygote which is fertilized totipotent, or which cells during division are still totipotent, we come up to the more or less stage where the cells more or less are still totipotent, but decreasing and becoming more and more pluripotent. So again, totipotent cells can form everything, including the placental parts. The pluripotent stem cells, as you would find them in a blastocyst, as inner cell mass or epiblast cells, only form the tissues, all tissues and all cells of the animal or of the organism. If you take out epiblast cells from the blastocyst and put them in a culture dish, you can culture them basically forever. These cells are then called embryonic stem cells, with the advantage that you can form an animal with those if you put them back. I'll come to that in a moment. So to the very right, you can see a colony of a number of pluripotent stem cells of a mouse in this case, which can nicely be identified. So once again, pluripotency means a cell of the epiblast or a cell can generate all cell types of the organism, of all 3 germ layers. Exception are the extraembryonic tissues, for example, a placenta. So how do we know if a cell is pluripotent or not? That's something, of course, we have to know. It's just not a textbook definition. It comes from experiments which have been done. So how do we know that? The best experiment is if you can produce an animal, a living animal from pluripotent stem cells or embryonic stem cells. So you take a blastocyst and inject test cells in there, which you somehow have labeled genetically or with a fluorescence protein. Inject these cells into the blastocyst, and to the right you can see such an experiment. Embryonic stem cells were injected, and these embryonic stem cells or the cells express GFP, green fluorescent protein. So you can see the fluorescence below here. Yeah. And when you transplant that or put the blastocyst back into a foster mother, the animals the offspring which come out are chimeric animals, because the animal is composed of the cells you put in plus the cells which already were in there. And if you cross those with each other, you then get, with a certain probability, chimeras which are only derived from the cells you put in there. So it's tedious work. What you can also do to speed up things is to do so-called tetraploid complementation. So what you start with is a blastocyst which contains 4N, which is 4N and not diploid, it's tetraploid. And such cells don't contribute to the animal at the end, because the animal wouldn't survive. So inject your diploid cells into that tetraploid blastocyst and put it back into an animal, and the only cells capable of forming the animal are the diploid ones. So you end up with an animal which is purely composed, or mainly purely composed, of the cells, the test cells you put in there. And if you get an animal out of the cells you injected there, you have proof that you had pluripotent stem cells injected. But there are other means to test whether you have pluripotency or not, because you can't generate an animal all the time. It's for, of course, a number of reasons which I think everybody understands. Also animal protection, of course. So what pluripotent stem cells do, as I said before, they can differentiate towards all cells of the body. So if you transplant those into an immunodeficient animal, immunodeficient mouse, they form tumors, for example, directly under the skin or in the kidney, part of kidney. So they would form teratomas, and these are tumors which contain all different types of cells of all germ layers. And that's the definition of a teratoma. So endothelial, mesoderm, endoderm, mesoderm, and ectoderm, all parts. But of course, again, it's tedious, and when thinking of animal protection, it's of course something you don't want to do regularly or avoid where possible. So there's another way how to test by looking at the transcriptome being expressed. So if you look at pluripotent stem cells and differentiate, you should find transcription activity, which is typical for the differentiated cells of the 3 germ layers. That's what's shown here. So you would like differentiate for 7 or whatever number of days your pluripotent stem cells, do a transcriptome analysis and find a typical pattern associated with mesoderm, endoderm, or ectoderm. So of course it's not it's kind of a functional assay, but it's indirect, but sufficient enough in the meanwhile because we have enough experience to tell whether you really have started with a pluripotent cell and that you have pluripotent stem cells at hand. So again, just to repeat what I said before, the potencies, the pluripotent stem cells can develop towards all 3 germ layers. Multipotent stem cells or tissue stem cells are resident in all tissues and regenerate the local tissue. Like you have them in the brain, you have them in the muscle, you have them, of course, in the bones for blood formation, bone marrow cavity. This is just another picture underlining what I said before. Neural stem cells produce neurons. Hematopoietic stem cells are blood-forming stem cells, produce all types of blood cells. That's the definition. Mammary or gland stem cells produce glands. So everywhere in the body you have niches which contains tissue stem cells and a low number, of course, of stem cells because you don't need many of those for regeneration. And I'll just get a little bit more into detail into 2 types of niches and stem cells. One is the intestine and the other is the hematopoietic system in the bone marrow. So the intestinal stem cell niche is within the gut, of course. In the small intestine you have, if you do look into the microscope basically and look at a cut or a stained cut, you find these typical villi structure and crypts. And at the lowest part in the crypts, you find those stem cells producing the epithelium. So directly at the lowest part of the crypt, you have single cells, so-called crypt-based columnar cells or stem cells. The hallmark of those cells is that they express a surface marker called LGR5. So these cells are LGR5 positive, and they are surrounded by Paneth cells which compose the niche and influence the activity of the stem cells by direct cell-cell interaction, by soluble factors. Wnt signaling is a very important one with countergradients of homomorphogenic protein gradients. So this is a blow-up of the crypt. Again, as I said before, you have the Paneth cells which compose up the niche. You have direct cell-cell interaction and a classical molecule involved in the instruction of neighboring cells by direct interaction is Notch signaling, which of course also plays a role here and is depicted, but also of course many other molecules, EGF, BMP, Wnt signaling, as I mentioned before. When stem cells start to proliferate, they divide, and the daughter cell, which later on differentiates and moves up the gut epithelium, rapidly proliferate. And these cells are called transit amplifying cells. They rapidly proliferate and then finally differentiate towards whatever the function is in the gut: tuft cells, goblet cells, enterocytes, endocrine cells, and so on. So that's one of the typical things you find in stem cell systems. Stem cells don't divide very often. Most of the time they rest and are genetically protected because of that, because the proliferating cells have to replicate the DNA. There's a danger that mutations happen. They're sensitive to mutations. So the stem cells as such divide seldom, but the daughter cells then rapidly proliferate and produce the amount of cells you need for a certain function. And in the hematopoietic system, you would call them short-term cells, but we'll come to that in a moment. In general, the general expression are transit amplifying cells. So again, how do we know? How do we identify the stem cells? One of the classical methods to do so is lineage tracing in vivo. So again, the question is, does your test cell, which you label, possess the full potency for regeneration? And what you usually do, or classically do, is you use a genetic reporter system driven by a tissue-specific promoter. In the best case, what you test, stem cell promoter. In this case here, the tissue-specific promoter drives the expression of a recombinase called CRE. And this CRE molecule in this case is fused to the tamoxifen binding or hormone binding domain of the estrogen receptor. Okay, another locus, another site, you have integrated something which responds to the recombinase, which can be recombined by the recombinase, and these are LOX-P sites. So in this case, in the mouse, integrated into the ROSA26 locus, which is constantly being expressed, transcribed. You have two LOX-P sites, which the CRE recombinase can recognize and recombine, and a stop codon in between. So nothing is being expressed as long as the stop codon has not been removed by the recombinase. So once the recombinase is expressed, because of the tissue-specific promoter, it cuts out the stop codon, and the gene behind, a fluorescent protein gene, encoding gene, or LAC-Z gene here, is then being expressed. You can stain or visualize the cells. So in this case, the cells would become green and blue as soon as the recombinase is expressed and activated by addition of tamoxifen. So this is how it looks like. In reality, you find LGR5, in this case, was being used as a stem cell specific promoter. You find at the bottom in the crypts green cells, which obviously seem to be the stem cells. So the question is, would these stem cells regenerate all of the epithelium? Well, as long as LGR5 is active as a promoter, you get GFP positive cells, which are green. But once LGR5 is turned off, because it's turned off as soon as the stem cell starts to differentiate, those cells, the green, would vanish, would be gone. But because of the estrogen receptor, because of CRE1 fused to the estrogen receptor, the LAC-Z gene is turned on constantly because the stop codon is being removed. So all of the daughter cells continue to express LAC-Z, and all of the daughter cells then would be blue. So if the stem cell is activated, if the stem cell produces all of the epithelium should become blue over time. That is what you can see here on this picture. Those cells which were green or blue at the beginning produce all of the epithelial cells, proving that you were looking at a stem cell. Another possibility since a few years is ex vivo in the dish to produce so-called organoids. Stem cells as such are able to proliferate more or less endlessly or for a long time. So if you take from the intestine LGR5 negative cells, sort them by flow cytometry, or enrich them, and take LGR5 positive cells, put them in culture, what you find is that those cells which are LGR5, only those expressing LGR5, can form colonies, continue to grow, can form colonies in a dish, and these colonies or these aggregates contain components which you would find in a gut. They start to differentiate and build little organs, basically. That's the reason why they are called organoids. And this is depicted down here. So it's just a staining of LGR positive cells and the cells as such. They build up lumen, they build up crypts, they have a villus domain, they look like little gut structures. So how does it, the second example is the hematopoietic system. So how does the system work? How do stem cells look like in the hematopoietic system? Well, what you all know is that stem cells, of the blood-forming stem cells, are in the bone marrow. And these stem cells are associated with endothelium, with vessel structures, with the endothelium. And what you can see here on the right is that you have a hierarchy. So the hierarchical system is you have the so-called long-term repopulating hematopoietic stem cell, or LT-HSC, at least in the mouse system, but it's very, very similar in humans. You have, when they start to differentiate, short-term repopulating hematopoietic stem cells, meaning they are still stem cells, but they don't behave like stem cells forever. They get lost over time. But still capable of producing all cells, all blood cells you have. So-called multipotent progenitors. Again, it's a progenitor, not a real stem cell. These cells get lost over time, but are still capable of producing all types of blood cells. And then everything splits up. You get a restriction towards the lymphoid line or the myeloid line, or some in part in between, so-called common lymphoid progenitors, clips, or common myeloid progenitors, which then continue to differentiate and produce the terminally differentiated cells, which overtake a certain function or fulfill a certain function, which we see in the peripheral blood, and I guess you all know. So the development of hematopoietic stem cells, or of the hematopoietic system, starts in the yolk sac. In the mouse, that's at day 7. The development is very, very, very similar in humans and all the mammals. But these yolk sac, so-called blood islands, don't produce all types of blood cells. They are not adult type of blood cells, but they produce primitive oxygen carriers and also endothelial cells to a certain extent. So the first real adult type of blood stem cells are formed in the dorsal aorta, also called aorta gonad mesonephros, or AGM region, which you can see after 10.5 days during mouse development. It's shown in the middle here, with the dark blue being the fetal liver and the AGM region. So this AGM region produces the very first stem cells, which then undergo a trip to they migrate via the vessels to the fetal liver, where they just expand a lot, and at the end of development, short before birth or around birth, they migrate to the bone marrow, where they then end and throughout whole lifetime as the adult hematopoietic stem cells, producing all of the blood cells. So again, this is a blow-up of the dorsal aorta, where the very first hematopoietic stem cells are being formed. They're formed on the ventral floor of this dorsal aorta structure and are formed out of a specialized cell which appears to be endothelial, the so-called hemogenic endothelial cell, or hemogenic endothelium. These are cells which completely transit from the endothelial morphology into a cell which is a suspension cell and is just like, with circulation, carried to the fetal liver, where they then expand. And this is just a label at the bottom of the slide showing a staining of these hemogenic endothelium cells and the first adult hematopoietic stem and progenitor cells being formed. So again, the question, how do we know that these cells are stem cells? Yeah, we're not just like trying to tell you textbook knowledge, but also trying to explain the experiments, the crucial experiments being done, which you can repeat, showing that this is a stem cell. So proof that you have a definitive adult type of hematopoietic or blood-forming stem cell is a transplantation experiment. But the pre-experiments just showing, well, during development, when do the very first hematopoietic stem cells form, was done in the 90s, the early 90s, by Albrecht Müller, a German scientist, and Alexander Medvinsky and Elaine Giajak, where they, unfortunately one has to say, but they chopped up embryos during certain stages of development and took different areas of the embryo from the yolk sac, from the AGM region or dorsal aorta, liver, and other parts of the body, cultured them for a short time in vitro, took the cell suspension and transplanted that into irradiated mice, similar to any type of bone marrow stem cell transplantation experiment, and looked after a certain time, well, does the mouse reconstitute? Does it form blood cells? Does it survive? And also looked at the spleen, because one of the first places where these cells end up are in the spleen, where they start like proliferating and forming colonies. So they looked at the so-called colony forming unit spleen, CFUS. The spleen, when they are isolated, look bumpy. You'll see that in a second. So this is how it looks like. So using a yolk sac, you see some bumpy structures in there with a control without anything. The spleens are smooth. The AGM region is full of those. And looking at long-term repopulation, meaning that after transplantation of a mouse, you find blood formation for the rest of the life of the mouse. They only got long-term repopulating hematopoietic stem cell activity, LTR, HSC activity, when they used cells of the AGM region. So most of the mice which were transplanted with cells isolated from the AGM region repopulate over time and generated blood, not from the yolk sac, not from the liver. The liver is not a de novo hematopoietic stem cell forming organ. Yeah, the liver is just a place where they expand, and body remnants also. So the AGM is the very first place where adult type of hematopoietic stem cells are being formed. So again, the tree as we see it today, it's in details reshaping now and then, but you have a hematopoietic stem cell over here, which forms short-term hematopoietic stem cells and then splits up, which would be the transit amplifying cells, yeah, multipotent progenitors then, forming clips, common lymphoid progenitors, common myeloid progenitors. And what you can see here is also the expression CFU gem, for example. These are the tests you use. If you isolate a progenitor and put it in culture, those cells would form a colony, similar to if you put a bacteria in a culture dish and see a colony forming. So similar to that, we'll find a colony forming which contains different types of cells. So if that progenitor cell only forms macrophages, for example, it would be a colony forming unit, CFU, M. If the colony forms granulocytes, erythrocytes, or red blood cells, megakaryocytes as progenitors for the platelets, and macrophages, you would have a multipotent cell, which is early up in the tree or far up in the tree. So the colony forming unit GEM is a very, very primitive type of cell, meaning that it can form all these different types of cells. So downstream you have, for example, this colony forming unit E producing erythroblasts and erythrocytes at the end, or CFU GM, colony forming unit GM. So that these are tests we can do to prove that we have progenitors in there, but it's not a real proof for stem cell activity. Yeah? The ultimate proof for stem cell activity, blood forming stem cell activity, is the transplantation experiment. Knowing that the stem cell as such can live almost forever and forms all of the blood once it has migrated to the bone marrow, settles down there in the niche, and behaves like a stem cell, of course, that's the real target you want to go for if you want to treat a disease in the hematopoietic system just once, by transplanting, correcting that cell, transplanting it, and correcting or curing the disease. Our current standard for blood stem cell transplantation is you have a donor which is not the recipient, so it's not the same person. Now you have a different and matched unrelated donor. It could be from the family, it can be a complete foreign person, another person, which is matched to a certain extent based on MHC or HLA, human leukocyte antigens, and other criteria, but it's always associated with the risk of rejection. The immune system, in both ways or both directions, recognizes the organ as, or the organism as being foreign and starts to reject that. So for that reason, we have to, in the clinical routine, do a lot of investigations before, a lot of tests before we transplant such cells or use them for cell therapy. The same happens if we transfuse just cells which survive for a short time, but they're also recognized by the immune system. So we characterize a certain number of antigens of those cells. Let it be stem cells or let it be whatever you transplant. You want to know if the recipient already has formed antibodies against that to avoid a rejection against certain antigens, so-called alloantibodies, which are directed against foreign antigens. And at the end, you do a so-called cross-match. You look or test whether a certain tissue from the donor, certain cells from the donor are rejected by the recipient or not. So for one certain donor, you test that with the recipient you want to give it to. At the end, what we want to have is to avoid rejection reactions at all, which can be deadly. You would like to use the patient's own so-called autologous cells. That will be highly desirable for obvious reasons. You wouldn't have rejection reactions. Using adult blood-forming stem cells is a challenge because the frequency of those in the bone marrow is very, very low. Only one out of 100,000 nucleated cells in the bone marrow is a real blood-forming stem cells. We do know a lot of the immune phenotype of the antigens expressed on the surface of those cells so that we can enrich them. And this is a paper from some years already where from a mouse at least, but also from humans in the meanwhile, these cells have been nicely defined. But in routine, in the clinic, we still just use the surface antigen CD34, which contains many progenitors also, and is not a pure stem cell population. A big challenge still is that we are not able to expand the hematopoietic stem cells in the culture dish. Once you take them out of the body and put them into a dish, they differentiate. And until today, there are no good conditions out there to expand them, a little bit maintenance, but this is still it's a holy grail, basically. And real gene targeting, so homologous recombination, gene repair, is, despite the advent of CRISPR-Cas methods, relatively inefficient still. So in contrast to using adult stem cells, which are in the body only in very, very low numbers, one alternative would be to use an upstream type of stem cell, the pluripotent stem cells, which, as I said before, can be nicely cultivated, not only of mice, but also of humans and of other animals. And the nice thing is that these embryonic stem cells, or pluripotent stem cells, are basically in a frequency up to 100%, or almost, if you know how to culture such cells. They show extensive self-renewal. You can keep them for decades in culture. You wouldn't want to do it practically, but in principle you could do so. And there are lines out there which are in culture since many, many, many years and still are functional. Homologous recombination is feasible for these cells. Mario Capecchi, for example, got the Nobel Prize for that. And these cells can differentiate towards any type of downstream stem cells you want to have them, in principle. So you could think of taking these embryonic stem cells, differentiating them towards hematopoietic stem cells, which then are multipotent stem cells, which wanted further stay in the bone marrow and go on further to differentiate towards effector cells. But of course, we know that all that the work with embryonic stem cells is highly controversial. Not with the mouse stem cells, with the embryonic stem cells, but with human embryonic stem cells. Especially here in Germany, we had it's very, very controversial, but of course in many, many other countries also. So work was prohibited, and Wolfgang Wagner's lecture the ethic lecture, will discuss with you, of course, many aspects of exactly this, these challenges and these hurdles, and the regulations, of course, which make sense. So some years later, Shinya Yamanaka and Sir John Gurdon showed that you can reprogram adult cells back into the embryonic state, looking as if they would be embryonic stem cells. And these cells were then called induced pluripotent stem cells, and they got the Nobel Prize for this. They showed that for mouse cells, and just 1 year later also for human cells. That opened many, many, many possibilities. So what's happening during the reprogramming process? Well, just imagine a mountainside or a hillside. So you have valleys where certain cells just drop down. They can just go down this certain one valley and become, say, erythrocytes. It would be a hematopoietic stem cell. It can do nothing more. It could also differentiate towards granulocytes, producing neutrophils. Okay? If you look into a different valley in the same at the mountainside, you would have a neural stem cell, which only can differentiate towards neurons. Terminally differentiated. So if you would want to have a pluripotent cell, you would have to go up all the way up the hills. And that's what they did. They were able to reprogram fibroblasts, which go all the way up the hill, and there could either decide to go down the one valley or the other valley to become neurons. And that's what they showed. So based on the method of Yamanaka and Gurdon, or Yamanaka basically, you could take differentiated cells, say from the patient shown here, take skin cells or whatever cells, reprogram that back to induced pluripotent stem cells and do gene editing with that, repair the defect if you know what the defect is. In this certain case, probably there are too many defects, but don't talk about that part. And use those corrected cells to produce, for example, hematopoietic stem cells or any other stem cells which you can transplant, or in the case of blood stem cells, go on further and produce erythrocytes, platelets, T cells, genetically manipulated cells like CAR T cells you hear about in a different lecture, and use them for transfusion or transplantation with the advantage that those cells are not being rejected because the donor is the recipient and you don't reject your own cells. Using that technology, you could also think of generating off-the-shelf cell products. So you would take a few donors which are genetically well-defined and would fit to many, many, many persons or patients in a certain population, reprogram that back to iPSCs, induced pluripotent stem cells, do genetic modification, expand them and freeze them in a storage bank as a master bank. So whenever you have a patient in need of such cells, you would identify the one fitting best, take that cells, for example, from a patient who has immunized against that or whatever, identify a compatible cell clone, thaw, expand that cells, do quality controls, whatever you want, and use those compatible cells. And as I see at the moment, it's Dinglish, so compatible, of course, is with C, written with C and not with K, and use those cells for the recipients. So you have one donor or a few donors which can be used for many, many recipients for an off-the-shelf product. The challenge we have now is not to reprogram cells to iPS cells, but to direct the differentiation of those cells towards the cell type we want to have. And for that, we have to know what the program is ongoing. So for differentiating the cells towards the pluripotent stem cells, towards the cell type we have to understand the hallmarks of the differentiated cells, of the genetic programs turned on or turned off in those cells, and direct the differentiation by leading them down that pathway and turning on and turning off certain genetic programs. So what you see here is a tree where single cell transcriptomics has been performed of a developing zebrafish. What you find at the end of the tree are the different cell types, all the different cell types you find in the body. And what you find also are certain places where that's literally saying that the cells turn either to the right or to the left, turn on a certain program. And those are these nod points or nodes, which are especially interesting because they direct whether the cell takes a turn to the left or takes a turn to the right. And where we want to end up is hematopoiesis, for example, which is down here at this end. So what we would like to find out is how to direct it there. And what we can do is what we have to know about is niche factors. So factors like cytokines directing differentiation of the cells towards a different path, which is difficult to control because you have gradient effects. You have to know which cytokine is where at a certain time in space and so on, and space. That's difficult to control. But we know a lot about the genetic programs turned on, as I said before. And the key transcription factors involved in turning on or turning off these programs. And when it comes to hematopoiesis, we know, for example, a number of transcription factors called like GATA2, RUNX1, FLY1, and so on, HOX genes, which we could overexpress in those cells or manipulate their expression to direct differentiation step after step or stepwise down to the cell we want to have. It's difficult, but it's doable. We just have to understand developmental biology better and for directing the differentiation. And we are having huge leaps in that direction. So for example, these are just a few of the many, many publications out there showing that we are able to produce megakaryocytes, so progenitors of the platelets, using pluripotent stem cells, for example, in this Nature Communications paper by chemical defined forward programming. And we are already also at the point that at least platelets which have been produced by iPSC differentiation, so induced pluripotent stem cells differentiated towards platelets, are already used for autologous transfusion for clinical studies, at least safety studies and so on. Phase 1 are safety studies. And with that, I'm finished for today with the stem cell lecture. I hope you learned a little bit, and thanks for your attention.